Hello everyone. Welcome to Radio Khachi 90.4 FM Hum Sabka Radio. This is Dr. Soni Kumari, Assistant Professor, Department of Botany, PPK College, Pundu. Today I am going to give a lecture on cell cycle and cell division. This topic is beneficial for the students of biology, both botany and zoology. This topic comes under the syllabus of BSc Sem 1, Botany Honours Papers CC2 and GE1. The reference books are Cell and Molecular Biology by P.K. Gupta, Cell Biology by C.V. Pawar, Instant Notes in Genetics, etc. Before starting this topic, I want to give you a little information about cell. So, what is cell? Cell is the structural and functional unit of any living being. So, we can say a cell is the smallest basic part of any life form. On the basis of number of cell, an organism can be unicellular or multicellular. Unicellular organisms are those who complete their life in a single cell, whereas multicellular are made up of multi-number of cells. These multicellular organisms also start their life from single cell. After getting several divisions, they become multicellular. So, how is it possible? For that, we have to understand cell cycle and cell division. Now, let's start the topic cell cycle. Cell cycle is the series of changes that occur in newly formed cell which involve in growth and division to form two daughter cells. We can also say that cell cycle is a sequence of events which a cell performs for its division. Let's understand the events of cell cycle. Cell cycle includes interphage or mitotic phase. Interphage is the largest phase of cell cycle. Previously, scientists called this phase as resting phase. They believe that cell is in resting condition because in this time nothing is visible from outside, nucleus seems inactive. But interphage perform the metabolic activity and prepared cell for division. It synthesizes RNA, protein, ATP and stored all the essential food materials which will be required for cell division. So we can say it is a preparatory phase. Interphase takes 95% of total generation times. Interphase is divided into three subphases. Number 1, G1 phase. Here the letter G stands for gap. So this phase is called gap 1 phase or first growth phase. This phase corresponds to the interval between mitosis and initiation of DNA replication. That's why it's also called pre-DNA synthesis phase. In this phase, intensive cellular growth and synthesis occur. In this phase, cell synthesize different type of protein, RNA, ATP and enzymes. DNA polymerase also synthesize in this phase, which will be utilized in the next phase of cell cycle. In every phase, cell prepare itself for the next phase. Now the cell enter into S phase. S phase, letter S stands for synthesis. So this phase is called synthesis phase. In this phase, cell double its DNA content This duplication of DNA is called replication. In this phase, synthesis of histone protein takes place. When we read cell cycle in various books, in some books it's written that histone protein synthesized in G1 and in many it's written in S phase. Both these information is not incorrect because in lower eukaryote 50% histone synthesized in G1 and rest 50% is synthesized in S phase, whereas in higher eukaryotes histone synthesized in S phase. These histone proteins take part in the packaging or coiling of DNA. From that, these long DNA can be fit inside the nucleus. If it is an animal cell, then centriole is also double. Here. Now the cell enter into G2 phase. G2 phase, this phase is called gap phase 2 or second growth phase. In this phase, synthesis of protein, RNA, ATP and spindle fibers takes place. All cell organelle double in this phase and the rest of the cellular synthesis takes place here. In plant cell, G1 phase is large while in animal cell, S phase is large. After completion of G2 phase, cell enter into M phase or mitotic phase. About M phase, I will discuss in my next lecture. Here, I discuss an important topic that if a cell not going to divide after their formation, so what exactly happens there? These newly formed cells are small in size, so they enter into G1 phase for short period to increase their size and exit G1 phase to enter into an inactive state called quiescent stage or G0 or G0 stage. Here, inactive word is in reference to division. Basically, cells perform all their metabolic activities except division. These cells become permanent and specialized in function called dedifferentiation. This is a suspended stage of cell, but when it gets any induction for division, it enters cell cycle. For example, parenchymatous cell in secondary growth of dicot stem form cambium after division. This condition is termed as dedifferentiation. The event of cell cycle is controlled by the molecule called regulator of cell cycle or cell engine. These are CDK and cyclin. These are protein formed and known as trigger protein or U protein. Leland H. Hartwell, Tim Hunt, and Sir Paul M. Nurse discovered these regulators of cell cycle. Number one is CDK, cyclin dependent kinase. CDK binds with cyclin and activates or inactivates the target protein to perform coordinate entry into the next phase of cell cycle. CDK are of 7 types: CDK1, CDK2, CDK3, CDK4, CDK5, CDK6 and CDK7. Number 2 is cyclin. Tim Hunt discovered first cyclin in 1980s. Cyclin are protein formed and degraded during each cell cycle. They were named cyclin because the level of these protein vary periodically during cell cycle. They are of 3 types. Number 1 is mitotic cyclin, found in M phage. Number 2 is S-cycline, found in S phage. Number 3 is G1 cycline, found in G1 phage. CDK and cycline form a complex called CDKC complex. This complex performs a common biochemical reaction called phosphorylation. When the cell goes through the cycle are arrested at specific point, these are called checkpoints. Checkpoints are the control system which control breaks and feedback signal. Break can be stop cell cycle at checkpoint and feedback signal delay the events of cell cycle. This checkpoint check whether the preparation are as per requirement or not. They are three checkpoints. Number 1 is G1 checkpoint or G1/S checkpoint. This checkpoint present towards the end of G1 phase called G1 checkpoint. In higher eukaryotes, this point is called restriction point or R point. In yeast cell, these are called start point. So, what are the things which will be checked here? Number 1 is whether the DNA is proper or not. Is there any damage in DNA? Because in the next phase, DNA going to be replicate. Number 2 is all the raw materials, that is protein, enzyme, ATP, which utilize in DNA replication are synthesized in sufficient amount or not. Number 3 is activates cycline which initiate cell division. That's why we say if a cell complete G1 phase, it's committed to cell division. Now the second checkpoint, G2 checkpoint or G2/M checkpoint. This checkpoint present towards G2 called G2 checkpoint. In this checkpoint, the things take in S phase as well as in G2 phase are going to be checked. Number 1, whether DNA replication has properly taken place or not. Number 2, have the cell accumulated the sufficient amount of protein, especially tubulin, enzyme, ATP, because now they are going for division. If everything is normal, then cell enter into M phase and the third checkpoint is present here. The third checkpoint is spindle checkpoint or M checkpoint. This checkpoint present at the end of metaphase, and the thing is checked here that whether the chromosomes are arranged on the equator and the spindle fiber attached to the kinetochore of a chromosome. If the spindle fiber attached to kinetochore, then it enter into anaphase and the cell division is going on. That's all about the cell cycle. My next lecture will be on M phase or mitotic phase. Till then, stay home, stay safe. Thank you.